as you make your way back from coffee, we'll go ahead and get started with our second session, our two keynote presentations. It gives me great pleasure to introduce Dr. Caren Goldberg.
Well, I wanted to thank Bob and everyone here for the invitation, and it's great to see so many people interested in eDNA, and excited about digging in to get to this sort of these standardization issues and how do we bring eDNA from where it is now into real acceptance. So amongst lots of folks. So I'm excited to be here. I'm excited to see all these folks. So we know that there's a lot of challenges with using eDNA detections for conservation and management. I'm going to focus on what I think of as the three main challenges. Maximizing detection, so how do we get our sensitivity up? And that means, as I think it was Caren Helbing mentioned this morning, that means that we're pushing our reactions really hard. Right? So to get the sensitivity we need, we are running things longer and harder than we do in other applications. And that means that we really need to focus also on minimizing contamination, because high sensitivity means even very tiny bits of contamination are an issue in a way that they're not when we're doing population genetics and other high quality tissue sample applications. And the third one, which we've already talked about a little bit today, but I'm going to hit on a lot more, and I suspect we're going to hit on a lot more all day, is interpreting uncertainty. So even with super rigorous quantity control quality assurance, there will still be uncertainty because we'll get these detections that are 1 out of 6, 1 out of 8, 1 out of 3. What do those mean? They might be slight bits of contamination. They might be critically important information about a super rare species. So what do we do with that? We'll talk about that. But I want to start out by talking a little bit about how I got here, which is, I think, the way a number of people, especially from the field side, get here, which is that although Jared characterized me as a lab biologist, actually I'm a field biologist who started to do work in the lab because I spent a lot of time doing a lot of field work for rare amphibians, and I know that surveys can be difficult, they can be destructive, they can be time consuming, and even with all of our efforts, we still may not detect species when they're present. Right. And when this paper first came out, the Fisatola paper in 2008, I remember the moment in which I heard about this work because I was so excited that we could do this. Even though I worked in a lab already that dealt with hair samples and scat samples and all kinds of terrible quality samples, it had never occurred to me that you could just look for DNA in the water to find the species that I was interested in, which was rare amphibians. So since then, I think we all know that eDNA detection of macroorganisms as a field has really taken off. There's sort of this lull. Let's see, I don't think you can see my pointer, so yeah, there's sort of this lull after the first paper came out. I think there were those of us who were thinking this is going to change everything, but a lot of people thinking this is crazy and probably doesn't really work. But of course, it turns out it really does work. And so after that lull, it's really taken off. And so this is, as of last month, we have already over 100 papers this year published.
If you need a pointer.
Oh, thanks. Great. The first set of species that I worked on was Idaho giant salamanders and tailed frogs. And these were great species to start on in a number of ways that I really didn't realize at the time. So first of all, these were in clear streams. So these are beautiful headwater streams, and so we could get a lot of water through those filters. And stream water, turns out, has excellent detection. I was super skeptical of working in streams. I thought it would be way better to work in wetlands, where I was thinking the DNA would just sort of accumulate and mix. But it turns out that streams have excellent places to catch eDNA. And the third part that I wasn't really thinking about was that these species are really different from every other species in their environment. And so they're phylogenetically distinct. And so I didn't have problems with specificity. I didn't have problems with filter clogging. I didn't have problems with detection. And of course, I thought, this is ready to go. Like, I've done it, it's ready to go. Since then, I have had the privilege of working on a ton of other species across lots and lots of systems, and I have learned a lot about what limits eDNA detection, what mistakes are made in the field. I have lots of partners who make all kinds of decisions in the field that I did not expect them to make, and I am going to talk today about some of the things that I've learned along the way. Some of them are in papers already, and I'll talk a lot about those, but I'll also talk about all the lessons that I've learned that don't end up going into papers because they caused a failure, and often those don't actually get out into the literature. So a lot of the lessons that I learned ended up in this paper, Critical Considerations. Now this was some work that I did with folks from around the world. It originated at this conference in 2013 for a symposium that I put together with some colleagues. And what happened was that we were getting together for this symposium, and we were all chatting, and we all had sort of the same concerns. So this is back in 2013. We were all doing sort of our first studies and running into our first big problems. And the concern was that most early eDNA practitioners were from non-invasive genetic labs or ancient DNA labs, where they either were doing this kind of clean work, which not everybody was doing, or at least they had the kind of protocols that I have in my lab, which is there's a lab that's totally separate that you have to shower and change into clean clothes to go into after you've been in the field, or especially if you've been near PCR products. Right? So it's a totally dedicated space. Now, a growing number of eDNA users at the time, and still now, were from more traditional population genetic labs. And as the field was taking off, I think people were just seeing it as like, hey, this stuff works, so I'm gonna do it in my regular workflow. And the concern was that these lack of precautions could lead to massive contamination issues. And like Mark Skinner pointed out, it could poison the well, right? So as a new field, we have to make sure that we're doing things right. And that's what led to that paper being put together. So we had a bunch of concerns when we started writing this paper. How do we write something relevant that doesn't quickly become outdated, right? It's this growing field, we're learning more each day. How do we write guidelines that are strict enough, but not so limiting that they're applied to reject papers inappropriately, right? So if we say this is how you do things, and we misspeak a little bit, we're a little inexact, that could be used as a rule to throw out stuff and prevent the growth of the field. And how do we make sure we're being inclusive of all kinds of different approaches in this growing field? So we had all those challenges that we tried to get at. And what we ended up doing was focusing on guidelines for lab separation and cleaning. So these are things that we could all agree on. And these are researchers from across the world. At the time this was news, but I hope that it's not news anymore, that standard PCR without sequencing when we're looking for targets is not specific enough for eDNA analysis. We need quantitative PCR or sequencing. We put in a validation procedure for new qPCR assays, some guidance for testing all samples for inhibition. I'll get back to that. Considerations for understanding, communicating uncertainty. I'll get back to that quite a bit. And then we have a discussion of spatial and temporal issues, limits with respect to estimating abundance. What we didn't include in this paper are specific recommendations for filter, for volume, for extraction techniques. I think when we first all got together, at least I thought that we would be able to do that. Like, we'll all just talk to each other, we'll decide what the best way is to do that. And what I learned pretty quickly was there is no one-size-fits-all design, right? People have different challenges, systems have different challenges, different goals, different levels of sediment, all kinds of different opportunities to get things right and wrong. And so there's really no one-size-fits-all design. eDNA is not one method, right? It's a thing that we can try to detect in lots of different ways and analyze. So these are some of the things that influence our limit of detection, right? When we design a study, we can choose to filter or precipitate. We can choose different filter materials and filter pore sizes if we're doing that. These decisions all influence each other because they influence how much sample volume you can collect, what your spatial sampling design is, and what materials you select really influence what the best extraction method is, and that's related to what analysis method you're using, and that's related to the number of replicates you need. These items, these elements cannot be pulled out uniquely to give a recommendation. This is a whole workflow that goes together, and lots of different labs have developed these workflows that work really well for them. But I think it's important to keep in mind, if we're talking about standardization, that we can't talk about these elements in isolation. because probably one lab has a really good reason why they're running that many replicates for that system with that collection method. And so these things all go together, which is a challenge for standardization. And there's a number of things that might influence why you make these decisions: your turnaround time, your number of target species, whether or not you need quantification, your field crew time and expertise, how scared you are that they're going to introduce extraneous DNA in there, the fine particulates in your system, degradation rates, transport, and then there are elements that have to do with the species that you're studying, their production rate, their space use, that influence what you should be detecting, or how you want to be detecting things. All right, so I'm going to talk about 10 lessons that I've learned in the field. I don't know what my timing is. What am I going to get? Okay. I'm going to talk about 10 lessons I've learned in the field, 3 lessons from the lab, some thoughts about standardization, and then that last piece, interpreting uncertainty. So, some lessons that I've learned from the field. Right, only bleach destroys enough DNA. That was something that I learned early on the hard way. Ethanol and flaming, DNA away, autoclaving, these are not thorough enough for eDNA applications. They do destroy some DNA, but we need to destroy all of the DNA. So forceps will carry enough DNA after ethanol and flaming and DNA away to contaminate all the samples after that. That's from experience. Okay, the flip side of that is bleach is really hard to clean out of grab bottles and other items. So once you have bleached something and gotten rid of all of the sample that you don't want in there because it's the last sample, it's then really hard to clean out of your next sample so you don't destroy everything that comes after that. So we do, the protocols that I've developed use a lot of single-use elements. People are very uncomfortable with that in the field. We go out with a lot of plastic, we come back with a lot of garbage. It seems antithetical to our goals as conservation biologists, but I just tell them it's better to create a lot of garbage and do the right thing than to create some garbage and have your data be garbage. So, to be blunt. So, other things that I've learned: filters that work great in streams may clog really fast in other systems. In the bottom corner here, we have a picture of the system that I work in Arizona. These are cattle ponds. They have endangered salamanders in them. You can't get more than sometimes 75 milliliters through the standard filters that I started out using. So increasing pore size, for example, to 5 microns will help with that. Right? So we lose some DNA when we increase our pore size, but it's more than made up for in these kinds of systems by the amount of water that you can filter. And that's in the old school style where we're filtering down and not the backpack sampler where we're pulling up where we could get a lot more water. Speaking of different kinds of pumps, we found that battery-powered pumps don't work better than hand pumps in that same system when we're filtering down. They're pulling the same, they pull the same volumes, right? But sometimes they're more convenient, especially if you have a system that doesn't seal very well and so you're pumping a lot with your hand pump. It can be more convenient. And that leads us to the next thing, which is that if you're using those filter cups, the seal is really important, especially if you're using a hand pump, because otherwise you're just going to spend a lot of energy creating suction that's just coming in through the sides of that pump. So those Nalgene filter funnels with the blue bases that have become standard in a lot of applications, they don't make a great seal. Right? So we've been using Sterilitex filter cups. Okay, back to sort of more general things. eDNA has limited dispersion in wetlands. This was something that really surprised me when I started with eDNA, and I probably told Jared when I first was saying how great eDNA was and how it's going to solve all of our detection problems, that you can show up at one side of a wetland and take a sample, and I thought that we'd get everything in the wetland. Right? Maybe not like a giant lake, but a wetland at least, but that turns out to not be the case. The DNA does stick around where the animal is. And probably the bubble of how far out you can detect an animal has to do with the degradation environment. How warm the water is, acidity, UV. Of course, it all actually relates to that bacterial community and how fast that eDNA is being degraded and from the animal. And then, of course, the space use of the animal. Is that animal dragging its DNA around for you and distributing it? Or is it a toad that just sits on the same rock every day and its DNA is just around there? Right, so we found this out in a few studies. This is the predicted probability of detection per sample of these Chiricahua leopard frogs down in Arizona. And what we found was, this was when we were just going to one side of the wetland. This is probably my second or third study. one side of the wetland, taking replicates. The larger the wetland, the less likely we were to detect the animal. We found the same thing. This is some work with Idaho Fish and Game, where they went around the edge of this lake. They took eDNA samples every 50 meters, and then they dip netted the whole thing. And there's a bunch of different codes here with the symbols, but mainly the green are eDNA detections, and the orange are field detections. And we mostly detected everything that the field crew found, and additionally we detected long-toed salamanders, which the field crew did not detect. But for toads in particular, we lost detections at about 50 meters from the animals. And that seems to be pretty typical across kind of wetland lentic systems. Even in flowing water, this is some of Taylor's data, and he'll be talking next, so you may see it again. Even in flowing water, transport distance is not that long, so it's contained to closer to the animal that is producing it, maybe within 100 meters or 1,000 meters. And our group has found this as well. This is these are data from sampling downstream of bull trout reds, and you can see that the eDNA signal in blue is really following the density of reds. So we're seeing these big spikes where there's higher density, and those drop off really fast. And this is something that goes with this other pattern that we've seen, which is that eDNA signal is quickly lost in flowing systems. This is one of the first experiments that I did with David Pilliod, putting 5 salamanders in a stream, sampling 5 meters downstream of that every few hours. And then here, right there, removal, nope. There, removal. Right, we pull the salamander cage from the stream, and the signal disappears within an hour. Right? I thought it would stick around for longer. And some work that Oregon Fish and Wildlife did last summer found the same pattern, which is that these foothill yellow-legged frogs, they tend to come out of the stream and bask. And so they're sampling, and they're actually looking at 10 frogs hanging out of the stream and not detecting anything in the stream. Right? So the signal's disappearing fast. Now you'd hope that not all of your animals are going to be out of the stream at the same time, but of course they're thermoregulating. So there is some likelihood that they all are going to be out of the water at the same moment, and you're going to fail to detect them when they're present. Right. eDNA detection has an optimal temperature. So as temperatures get warmer, we get higher degradation rates. So we see a drop-off in detection as temperatures get above about 25 degrees Celsius. This is down in Arizona where water gets quite hot. But we found when we were working in colder systems is that eDNA detection actually goes up with temperature, probably because animals are more active and they're putting out more eDNA. So we've got a balance between eDNA production and eDNA degradation that probably means that we have an optimal water temperature at which we can detect eDNA. eDNA detection varies by species, and this is pretty obvious that a frog would put out more eDNA than a snake or a turtle, but we've also found this to be true even within frogs. This is a system, this is Moses Lake in Washington. This is the last remaining northern leopard frog population in the state and in the whole area. This system also has bullfrogs, invasive bullfrogs, and what we found in this system was we detected bullfrogs at 77% of sites with visual observations, so good standard eDNA detection rate. 2% of sites with visual observations of the northern leopard frog. This is the worst I've ever seen detection, and of course it's a really important system to be doing a good job in, and I don't know why this is happening. They don't have the wrong mitochondria, if you're thinking that, because I checked. Okay, 10, eDNA production varies widely among individuals. This is a study that we did, but the number of people doing similar studies have found similar patterns. These are bullfrogs in little tubs. They're all fed exactly the same way. They're from the same source. They're the same age. And we pulled those bullfrog tadpoles, and before we assigned those tubs to different degradation treatments, we took a time 0 of DNA production, just so we could correct for the little changes, little differences between how much eDNA we were starting with. And what we found was some animals shed 2,000 times more DNA than other animals. Right? And they all looked the same. Everybody looked healthy. It wasn't patterned by shelf or anything like that. It's just some animals seem to be super shedders of eDNA. Okay, that's not totally field, but those are the Those are the more fieldy based lessons. 3 lessons learned in the lab. Primer specificity at least was much lower than I thought. Right? It's so this was one of the first designs that I did. This was for the Idaho giant salamander, 78 base pairs. I thought it worked great. This was conventional PCR. I ramped up the number of samples that I was doing, and I suddenly got a lot of false positives. Turned out it was me, probably, or some other human. So these were the mismatches, right? So in standard PCR and the kind of population genetics work I've been doing, if I designed a primer that had these mismatches, I would think this is not going to work. But we're pushing these reactions so hard for something so rare that might not be there that it can make all kinds of mismatches that we weren't expecting. False positive signals are pretty common. Now this is my workflow in my lab. We've estimated false positive rate at that binomial probability of 2%, 0.02. So that seems pretty low per well, but with 96 wells on a plate, that means 0.14 probability per 96 well plate. So when we're getting these 1 out of 3, 1 out of 6, there's some probability, non-zero, important probability, that that's a false positive, right? It's more likely to be a true positive. But sometimes it's a false positive. I mean, we should talk about that today. Inhibition is common. We've already talked about that. May vary among samples and among seasons. How much more time do I have?
You've got 20 minutes.
Okay. So we had some recommendations from the critical considerations paper. One of the most important ones was to do a pilot study that's implementing your whole protocol, making sure it's working in places where you know the species is, working in places that the species isn't, right? Giving you the right negative answer. Testing those extraction analysis protocols, validating those PCR assays in silico, so against that database and with enough standardization criteria that you have lots of different base pair changes. In vitro with tissue samples and in the field where you know the species is. Right? In the field, collecting negative controls, employing decontamination protocols, for all equipment and clothing. So I know in some systems you can sample without getting into the mud and the water, but in a lot of systems you're in there. And so you still have to be decontaminating between sites to make sure we're not moving DNA or disease. Collecting multiple samples at each site to address false positive. We can estimate those address false negatives and estimate detection probabilities. Right, in the laboratory, we have these standards to process samples only in that dedicated space with regular decontamination, using filter tips, using quantitative PCR if your target is just a few well-characterized species, or high throughput sequencing, metabarcoding if you have a lot of targets, using technical replicates greater than equal to 3. This was an element of debate amongst authors, how many technical replicates, and there have been papers on that, and like I said, it is probably very much related to the entire workflow, and then archiving samples, which Taylor's going to talk a lot about. And then we have reporting, right? So this is definitely something where we haven't done a lot of standardization across the field for. Reporting quantification values as copy number per volume sampled, right? And definitely my papers are guilty of not doing this and just using a tissue sample from the lab that we got a qubit reading on and using that. But really, that means we can't compare between labs or even between years. So using copy number, acknowledge our challenges, inferring across space and time, whether we're just grabbing presence, like presence of the eDNA sample versus presence of a viable population, confounding sources, is DNA possibly being brought in by something else? And maintaining an archive database with collection date and exact geographic location. So we had a bunch of discussion points within our group, right? We had people from all over the world, and we were trying to create some kind of agreement on how we should do this new thing. So we had this discussion point on how much lab separation is required, right? Is it enough to have separate parts of a single lab? That's how I was trained, right? Extraction over here, PCR over here. We agreed across the labs based on experience that this is a terrible idea for eDNA and it will lead to contamination eventually. So even labs that originally did not agree with this ended up going in that direction. But do you need a different building? Do you need positive air pressure so you're not like bringing in air that might have PCR product from the rest of your building? I think everyone agreed this was a good idea. when it's possible, but most of us do not have access to that kind of space. Do we need to be wearing booties and hoods and face masks? So these are used in some labs, especially when people are doing shotgun sequencing, when human contamination is an issue. Most eDNA labs do not do this. And in most eDNA labs, it's okay to get human DNA in there. I mean, we don't want to cross-contaminate, but in a lot of systems that I work in, people go swimming in there. So if my assays are going to accidentally pick that up, that's a problem, right? So here's the one that already came up today, right? Does one positive equal detection? Is that important or is that noise? Right? So this is, and here, this is an example of from one of my graduate students. This is one positive, really low level positive, coming up for a site that had one metamorph that they found. So this is probably a true positive, but we couldn't repeat it, and so it got characterized in our characterization of threshold values as negative. So does one positive equal detection? Most labs said no. You have to see it multiple times in order for it to be a detection. But some labs, especially the labs in Europe, said absolutely, one positive is a detection. We're looking for really low level stuff. And that's a detection. And if you're getting false positives, then you need to work on your workflow. You know, so we compromise. So in the paper it says if you can only see it once, you should view that with caution. But I think it's really a matter of how hard are you pushing that reaction and how sensitive are your techniques in the lab. You can see in our lab, we run to 50 cycles. A lot of labs run to 40 cycles. If we run to 40 cycles, we're gonna see less of this. So we might miss some true positives, but we also might reduce our false positive rate, right? How hard should we push these things to get, is it okay to be slightly into the false positive territory so that we can get more true positives? Or do we wanna back off? And so I think it's related to that entire workflow setup. We discussed what kind of internal positive control should be used. Do IPCs, so the ones that we get off the market or the ones that some labs make themselves, there's a question about whether they control for all kinds of inhibition or whether they're biased towards certain kinds and may sometimes come up and look great when your sample is actually inhibited. There's a suggestion that maybe concentrations of that IPC should match the expected concentration of your sample. Maybe what the companies recommend we run these at is really high compared to what our samples are, and so maybe that's not a good control. But the concentrations for these IPCs are proprietary when you buy them from a company. We don't really know, so you can dilute them down and kind of push that CQ value, but we don't really know. Last discussion point, I think, was how much sequencing validation is required. So we all agreed that if you're running only conventional PCR, then it's required for every sample. You can't just look at a gross band and go, that's about the right size, so I've got a positive. But for qPCR, we agreed that it was required in situ, so from the actual field sample for some samples. But we don't have a recommendation on how many samples. I tend to do it on the samples that are most iffy, right? The samples that are low level and the samples that we're not sure. It seems like a weird positive there, but it's important, right? So, so when assays are created, right, we can validate them in silico, in vitro, in situ. They're usually done by the originating lab, right? So we've gotten to the point where now we have a lot of assays that are developed and other labs want to use them, right? We don't want to reinvent the wheel. We want to have good assays. We want everyone to move forward and not start from the beginning. But the problem is, how do we compare the performance of different assays? So let's say, like we do for many of our invasive species, we have multiple labs that simultaneously developed assays. How do we decide if all of those assays are of equal quality, or if some of them are not as sensitive for whatever reason? And then how do we ensure that everything is standardized between labs, so when these assays move, how do we make sure that every lab is running that assay the same way? And I just want to say this one thing, because a lot of people ask me, where can I go in GenBank to find these primer sequences? Right, so of course that's not what GenBank is for. But also, I just want to say there should never be a database of just assay sequences that are decoupled from their conditions. Right? Assay sequences always need to be coupled with what went into that PCR and the conditions. So in order to remain validated, because if we move these primers, to a different system, they're going to be less specific or less sensitive or more sensitive than they were with whoever developed them. And I know that this is different than in my career before this, dealing with tissue samples. I would usually just take other people's primer sequences. I would throw out their assay conditions. I would use my own buffers and my own enzymes because I liked them better and they worked well for me. And I knew that they were okay because they produced the sequences, right? Because I was looking at them. We don't have that backup here. You're just getting positive or negative when you're using qPCR. And so, and this is specific to qPCR, where you're just getting that positive or negative, somebody else has validated it, so unless you have the tissue samples to revalidate that assay, you better be using their exact conditions. Right, so we should never decouple those. So in addition to making sure assays are specific, so they don't pop up for the wrong species, we can also document their sensitivity and accuracy with limits of detection and quantification. And I'm just going to talk for a couple minutes about this project that I've been working on. It's led by Katie Klimas, and it's just trying to standardize some definitions of what are these things, the limits of detection and quantification when it comes to eDNA assays, because these are defined for other fields, but eDNA samples don't fit into that very well because they're so low level. So we have our eDNA standard curve that we make, and that eDNA standard curve has a linear range. So we draw this line, and we can use this line to figure out if you have a sample along that line, what the quantification is of that sample. Right? But what happens at the low end of that, and the upper end is the low end, the purple ones, is it gets messy. Right? qPCR is not great at quantifying at these super low levels, and if we ever want to use those quantification data, and we can talk about whether or not that's a useful piece of information, I think that it is to a certain extent, then we have to figure out what's happening at that low level. Where do we go from it being noise to it being signal? Right, so can we reliably detect stuff that's that low? And then how do we quantify it? So limit of detection, limit of quantification. Right, so we want to be within this linear range when we're quantifying, not where it sort of curves over at that end. Right, so how do we define that for eDNA? So from the microbiology, food safety, diagnostics literature, limit of detection is the lowest standard concentration where you can detect at least 95% of positive replicates. So therefore we're confident that if this number of copies of eDNA is in our sample, we will detect it. 95% probability, you run a few replicates, you've got really high probability of detection. One of the things that comes up, I think every time we have a meeting about the limit of detection, is that this does not mean that stuff that comes out below the limit of detection is false and should be thrown out. This means we're not sure if something is that really low, we're not sure we're going to be able to detect it, but it doesn't mean that if we do detect it, there's something wrong with it. Right? So the limit of detection is just what can we be confident we're going to detect, and we can use that to compare between assays. Right? So I use that for my assays. When we generate a new assay, I use the limit of detection to figure out is it okay, is it sensitive enough, right? So so LOQ then, the quantification is the limit, is the standard concentration that can be measured with some accuracy. So we use the coefficient of variation of many replicates to figure this out, and there's a cutoff. This is from the literature of about 20 to 35% coefficient of variation. So very few studies describe limit of detection and limit of quantification for their assay at this point. In order to do it, you just need to run to about 20 replicates or more per standard. That LOD, LOQ calculation, it only has to be run once for your assay, right? And then assuming that your assay always stays with its components, even when it moves to a different lab, those things go with that assay, not with those primers, but with the assay as a whole, all the conditions, right? And we can use those to compare between assays and to make sure that all of our assays are having the sensitivity that we expect of them. And also figure out if we're going to be using quantitative values, are they noise or are they signal? Is everything below 10 basically just stuff that's below 10, right? Or can we actually use those in a quantitative framework? And Chris Merkes is working on an R package for this, and so we hope to get that out in the literature really soon. Right, so the number should hold as long as the assay is always run the same way. You don't have to do this over and over again. Okay, finally, interpreting uncertainty. So even with taking every precaution, we still have uncertainty, right? We can bleach everything, we can do everything right, really high QA/QC. We still have these low-level positives. What do we do with them? Right? So these low-level signals, they're either critically important, the most important thing that we're looking for, or they are totally misleading. Right? eDNA methods are highly sensitive, therefore super vulnerable to these slight contaminations. We also sometimes have these allochthonous sources, is sometimes called the wicked problem of eDNA. In my area, we have moose. They love wetlands. They travel from wetland to wetland. They probably bring a lot of eDNA with them, so they could bring a signal of an animal that is actually not present at that site. So I'm proposing that when we code our samples as positive or negative, that's really not the best way to be talking about these levels of evidence, right? It might not be the best way to communicate results. So we tend, in eDNA, we tend to err on the side of caution by reporting only positives that are confirmed, right, in multiple wells. Now, from what I saw this morning, that might not be universally true, but from my experience, that's what we tend to do. We tend to say we're only going to report it as positive if we see it a bunch of times, multiple wells. But we know sometimes real signals look like false positives. We know that sometimes it's a real signal, but we only see it once. Even the vast majority of the time that might be true. So depending on the application, a false negative result may be as damaging as a false positive result, right? So I was just at a conference where Brittany Mosher was talking about looking for BD in high elevation, that's the pathogen of amphibians, in high elevation ponds that they were thinking of reintroducing toads to. They had a lot of ponds to choose from, and so they wanted to know which ones were BD negative. And it was really important to them that even if there was the slightest indication that there might be BD there, that they know about it, that it doesn't get coded as negative, so that they can avoid that pond because they have a lot to choose from, right? So sometimes that can be as damaging. So, but how should we communicate that uncertainty so we don't just freak everybody out, right? Yeah, so there's a spectrum, right? We've got this one end, there's no wells positive at all, and of course this depends on how many wells you run, right? But sometimes we just have no evidence. That's pretty clear. Sometimes everything comes up strongly positive. Great, we're good. But a lot of times we're kind of in the middle, right? We have these inconclusive ones where at least one well is positive. I don't know if there's more levels of this, right? Is it just inconclusive, or are there multiple levels of inconclusiveness? And is that confusing, right? If we just start reporting to people this is inconclusive, is that going to be satisfying or communicative or just drive everybody crazy? And then we have some evidence, right? If you have maybe more than one well is positive, but not all the wells are positive. Of course, this really depends on how many wells you ran. Did you run 3, 6, 12? Right? So we need to decide, or it would be helpful to decide as a community, how are we going to communicate these results, right? I've been at meetings where managers are going bonkers because they're getting results that say things like slightly positive. And they're just like, what? What does that mean, slightly positive?
It's like slightly bright.
Right. That's what I say too. Yeah. So, so figuring out how are we going to communicate this uncertainty, because the choice right now of not communicating the uncertainty isn't going to serve us in the long run, right? Because when people delve into that, they realize like there is uncertainty, right? Better to be out front with what that uncertainty is and address it than have it be less not communicated. And yeah, there's a lot of people that have contributed to the work that I do and that I've worked with and learned from, and I just wanted to acknowledge all of them, including my students and technicians, and thank you guys for having me here.
We definitely have time for some questions. Jared.
On the terminology front, I like the use of the word, instead of inconclusive, which creates ambiguity and uncertainty and fear, if we use the word suspected, and that suggests that we should retest, relook again, dive deeper. So around terminology, false light, that word seems to be accepted more readily than already.
I feel like that really puts, sorry, I'm supposed to repeat the question, right? So, yeah, so Jared's saying that what they do is when something has one well positive or less than whatever threshold for a full positive, they send people the results suspected. And I think that really, it puts the weight on the fact that it really might be positive when sometimes it really might not be. So I see the benefit of it, but I also see the danger. I think it's a choice.
Yeah, the criteria are a little more detailed than that. It's not just based on the number of wells that come back positive. But basically we have a lab call, which is analyzing the technical replicate, and then we have a biologist call, which analyzes all 3 technical replicates together. And selectively,
Samples, right?
So based on that biological consideration, that's where we use the assignment of suspected. At the lab call is purely binary.
Yeah, so I'm going to summarize that a little bit for the video, which is that they're saying that there's multiple things going into that. There's a biologist call, they're looking at multiple replicates, multiple sample replicates, as well as multiple technical replicates before saying that things are suspect.
Yes.
Yeah, Caren.
Hi, Karen.
Hi, Caren.
I think that point is really critical because the inherent challenges, and I really loved your talk. It was just encompassed so many different aspects, and the complexity of detecting eDNA in all the variety of different forms that are going to be used, have been used, and stuff like that is amazing. And the thing about eDNA is that there really is that dichotomy of, the lab detection process, which is what everything is based on, ultimately, but it's intimately related to the field application, and those two have to integrate in terms of the application itself, and it has to integrate in the interpretation of it as well. And I think that in and of itself is the key challenge, and that's where we need to look.
Yeah, I'm gonna summarize that as what we do in the lab that everything is based on, and what our decision making is in the field, how we interpret things, how we took that sample, what it means within that system. That they can't be independent of each other. Is that?
Yeah.
Yeah, totally agreed. Yeah.
Just a quick observation. The bounds of what we're discussing mostly this morning is the parameter of conservation science and environmental monitoring. So aside from the lab level and the technical level, at the environmental monitoring level, cautionary principle of 1 in 3 or 1 in 6 to err on that side of caution and have that be a false positive. You want to lean that way in terms of conservation in case of your reintroduction of media in those systems. You could foul a restoration study, or as the talk this morning with Red Side Case showed, is that 1 in 6 detection cued in for DFO to go in and do heavily intensive conventional sampling, follow-up sampling, and the like. So I think bracketing the uncertainty around it is great and required, and the semantics of that. But never losing sight of the fact that 1 in 6 you want to lean forward on, depending on your conservation goals, that until you can prove conclusively otherwise, you tainted the sample or contaminated or those things. You want to be quick before you brush that off on a technical level and say, oh, below the threshold for so many replicates or whatever. There's something there that requires further investigation, like Jared mentioned, how we sponsor that, but that cautionary principle is first and foremost, I think.
Yeah, so I'm gonna try to paraphrase that as well, which is, no, it's great, which is that Mark's advocating that in these applications it's really important to err on the side of this might be a positive and not disregard that until we have evidence, right? So not err on the side of thinking it might be a positive, we can't, thinking that it's a negative unless we can repeat it, but err on the side of reporting that this might be a positive. So similar to what you guys were saying too. Yeah.
You mentioned in your presentation about commercial IPCs, but have you in your lab ever tried designing your own IPC assay?
We haven't. So we've used the commercial ones so they can just be, purchased, and yeah, we haven't put our efforts that way. Yeah. Yeah, Caren.
Just a comment again about the IPCs and the use of it. Those are really useful for detecting inhibition within a sample itself, but they do nothing to detect whether or not the actual sample that you collected is degraded or not.
Yeah, so Caren is pointing out that the IPCs that are generally used test for inhibition, PCR inhibition, because there's co-extracted stuff that you collected and is in your sample preventing PCR, but there's a whole other level that her work addresses with the looking for algae that would catch things like the time that they didn't rinse the bleach out of the grab bottles and everything came out negative.
Yeah, I think that's one of our big sources. We've seen error rates or fail rates with degradation of 10 to 25%, depending on the study, of our 45 studies we've looked at. And I actually do think that's one of the sources is people not rinsing properly when we're using the collection or grab bottles. So I also want to say really silly comment, but it's an awesome marriage. I got my two favorite Karens in one room.
So we should probably go ahead and cut the questions here. We've got time at the break, but we need to give ample time to our other keynote speakers. So a big round of applause for that. And again, it gives me great pleasure to introduce our second speaker, Dr. Taylor Wilcox.
I hope you'll excuse me. I seem to be losing my voice a little bit. It's just great timing for that. Yeah, I can try and do that. And it's really advanced. All right. So I'm really excited. Are you
Need a pointer?
Pointer? Excellent. Thanks. To be here because I'm really excited about the potential for eDNA to characterize patterns of biodiversity on the landscape. As I'll talk about here in a minute, I think that standardization is a really important next step for us to take as a community of eDNA users in order to accomplish this. Before I get started, I do want to acknowledge that some of the ideas I'm going to talk about today they don't originate from me, but they really come from the collaborative environment at the National Genomics Center for Wildlife and Fish Conservation, where I work, particularly with the scientists that are listed here on this slide, and in particular the range-wide bull trout project and the eDNA Atlas project that I'll talk about a good bit today. We're really spearheaded by Kevin McKelvey, Mike Young, and Dan Isaac, which sounds like some of you have already worked with them in various projects before, as well as many other people at the National Genomics Center, but as a larger group of collaborators that include folks from academia, federal government, state government, tribal governments, and nonprofit organizations. So I think we're all familiar with this allure of eDNA sampling, and that is that all organisms, from prokaryotes up to megafauna, are releasing their genetic material into the environment, or at least their genetic material is available for us to sample. And so you know that within your environmental sample, there's this potential to detect a wide range of taxa and to characterize the biodiversity that's there at greater resolution and over greater scales than has ever been possible before. But I think some of the talks that have already come illustrate this reality that we're also familiar with, that typically our sampling efforts are motivated by one or a couple of key focal species. So maybe this is an invasive species. We want to detect that invasion front and put an end to it before we have an established population. Maybe it's an endangered species. We want to find that remnant population so that we can protect it and its habitat. So my goal today is to draw some examples from the National Genomics Center for Wildlife and Fish Conservation that I think illustrate how when we have standardization in place and we're thoughtful about how we archive both our physical samples and our eDNA data, we can extract more of the biodiversity information that we have from our eDNA survey efforts than would otherwise be possible. So first I'm going to talk about the range-wide bull trout eDNA project, which really launched the eDNA program at the National Genomics Center in many ways. And then I'm going to talk about the newest iteration of that, which is the eDNA Atlas project. Then I'm going to talk about the Atlas in action. That is a case study where standardization, sample and data archiving were useful in bringing new biological insight into a management relevant question. And then I'm going to talk about the Atlas of the future, and that is what might archiving of both our physical samples and our eDNA data look like as a broader community going forward. And I want to emphasize that for this last point in particular, my goal is not to lay out kind of bullet point, this is what I think you need to do, but I really hope to jumpstart a conversation as a broader community of users about how we could do this effectively. So I'm going to talk a fair bit about bull trout. Bull trout are listed as threatened under the U.S. Endangered Species Act, and I may not provide this context. I'm in the U.S., and so that's the law that we're really thinking about. I'm not going to talk about SARA, but the U.S. Endangered Species Act is kind of the law that has teeth for us when we talk about imperiled species. So bull trout face threats from climate change and invasive species, and I'll talk about those threats in a little bit more detail. They face threats from habitat destruction and historically from overexploitation. And this is a species that naturally occurs on the landscape at low population densities, but they're really widespread. And so when you sample for bull trout using traditional methods, you need to put a lot of sampling effort on the ground over a potentially really remote, rugged country. And so they're a really good fit for eDNA sampling, which has high detection probabilities for fish and streams, and which has really rapid sample collection. And so that's why a few years ago we launched the range-wide bull trout eDNA project. And basically the goal of this project was to determine the distribution of bull trout in potential juvenile habitats, so these are cold, largely headwater streams, across the species distribution in the Columbia Basin. Let's see what my next slide is. And because bull trout are federally listed, we're able to engage the interest of a wide variety of collaborators. So these include federal agencies, but also state governments, tribal governments, nonprofit organizations, who helped to fund some of the analysis for this project. But importantly, they did the vast majority of the field sampling. And often the sampling that each individual collaborator is doing is more regional, is more focused spatially than the larger bull trout project, because they're interested in bull trout for their own specific reason. So you might have a national forest that's interested in bull trout just on their national forest. You might have a nonprofit organization that wants to know about bull trout in a watershed that they've been monitoring for years. So I'll give you one example of a motivation for which we might have a collaborator sample for bull trout. So we found that the U.S. Fish and Wildlife Service is prepared to accept eDNA data as evidence that ESA Section 7 consultation is not required. So this is a piece of the Endangered Species Act that says that if you're a federal agency and you have some management action that could impact a listed species, then you need to go through this formal consultation process with the U.S. Fish and Wildlife Service to determine if your management action can go forward or if you need to modify that action in order to minimize impact on your listed species. But in this case, what we found is that the U.S. Fish and Wildlife Service recognizes that if we're not detecting bull trout eDNA, then that's strong evidence that there's no bull trout population. In those cases, the Forest Service doesn't need to go through a consultation process in that system, and they can focus their efforts on other habitats where there actually are bull trout populations. And so we have many of these smaller sampling efforts, but but I think that this is a first example of how standardization can be valuable, because although we have dozens of different collaborators, each of those collaborators is using the same peer-reviewed standardized protocol for collecting their samples. So that in aggregate, we now have, I think it's about 3,000 to 4,000 different sites that have been collected, taken to the lab, extracted, and analyzed in the same way. So this is actually a screenshot from the range-wide bull trout eDNA project, and I'll have a link for that on the last slide, I think, where you can access all of this data. So you can go and see where samples were collected and what the analysis results were. You can download shape files if you want to do your own analyses with these. And this has been a really useful data set for us because now we have pulled together these individual collaborations into a larger data set where we can ask questions about what are the drivers of bull trout distributions at the landscape scale and how might different management actions influence those distributions in the future. And that's bringing us to the newest iteration, kind of building off of the bull trout model. So again, we have many different collaborators who are going out, they're collecting samples using a standardized protocol. Samples come back to the National Genomics Center. We do an analysis that they asked for, but then we're also putting that data up on a public database. So the data is kind of slowly moving into view as we get written permissions from all of our collaborators, but this represents at this time about 8,000 samples that have been analyzed for at least one of about 30 different species for which we do taxon-specific quantitative PCR analysis. And I think that this idea of having a public database where you put your eDNA data is pretty simple, but it actually helps us to unlock more of the biodiversity information that we could get to from our eDNA sampling efforts in a couple of ways. So I'm going to kind of try and flesh that out using a cartoon. So this is the relationship that many eDNA labs that have a service component have with their collaborators. So our collaborator, I'm not going to try to use the pointer, has some question, and so they're collecting samples in their region of interest. They send it to the eDNA lab, we do an analysis, and then we send them a report. And that report is certainly useful for the collaborator. It addresses their specific question. But that biodiversity information that we've generated largely becomes unavailable to the broader public and to the broader scientific community. Because that report may be difficult to access, and it's going to be really difficult to draw data across multiple reports to multiple collaborators generated over time so that you can get a more cohesive view. So simply by having a public database here, we're able to essentially intercept that biodiversity information and format it in a way that naturally aggregates data over multiple studies and over time. And it doesn't decrease our ability to provide that data to our collaborator. And now what we have is this kind of accumulation of samples. So maybe with the bull trout project being an example of this, you you have sampling in one area for a species of interest, and you have someone else who's also interested in that species, and they're sampling another area. So over time, you're naturally getting a more comprehensive view of that species over space. And then often we have cases where we've done an analysis in one area for one species of interest. Now we have a new project and we're interested in another species. And so now we're starting to build up a greater taxonomic view of what's there as we do add new species on top of each other. And then we expect for this database to accumulate data over time. And so we're starting to gain a temporal perspective. How are these communities changing in a temporal way? And then the less visible part of this, but I think just as important, if not more so, is that we're trying to be intentional about how we archive our physical samples. Because once we've extracted DNA in particular, done our initial analysis, that extracted DNA can be preserved potentially for hundreds of years. And it's pretty inexpensive for us to revisit that extracted DNA and generate a new analysis. And of course, when we do that, we can put that into this atlas, into this public database. So there's this potential when we archive to go back immediately and look for another species that it turns out we're actually interested in. It also opens the door to doing retrospective analyses. So the species that are of interest today are not necessarily the species that will be of greatest interest tomorrow. And so there may be species that we wouldn't think of to survey for now, but if they have a range contraction in the future, suddenly we're going to be very interested in where was this species historically. And if we have an archived sample, we can actually go back in time in a way and look for where that species was. And it also buys us access to future technologies that may be, well, I'm just going to be confidently say that are going to be better at generating biodiversity information with greater accuracy and reliability from our environmental samples. So next I'm going to talk about a specific case study where this is exactly what we did. We did a highly targeted species-specific survey effort. And then we had a new question that required new biological information, so we're able to access our archive and bring that data into this question. So this is building off of the bull trout project, and so I'm going to provide a little bit more context on bull trout. So a lot of the sampling effort that we started for bull trout was motivated by concern about where bull trout are going to be in the future due to climate change. I kind of alluded to this before, but juvenile bull trout in particular are tied to really cold water habitats. And as we're predicting those to drop out as climate change progresses, we're expecting for there to be shifts in the distribution of bull trout. So we want to know where are bull trout going to be in the future, where are they going to be in trouble, where might be strongholds. And so this is a pretty typical thing to do in conservation biology these days, right? You have an idea of where your species of interest is, and then you have some idea of what the environmental conditions are in space right now. So you build up a species distribution model. You have a climate model that tells you where that climatic niche is going to be in the future, and then you combine those two models to come up with a future species distribution. But increasingly we're seeing in the literature a reminder that species interactions matter. And so if you have some other species, then the one that you're looking at is not necessarily just responding to abiotic conditions, but those species interactions are going to be important to consider in your climate projections. So for bull trout, an obvious interacting species to think about are brook trout. So in our neck of the woods, brook trout are invasive species, and they have a high niche overlap with bull trout. They can also invade these cold water habitats, and there have been studies that found that brook trout can competitively exclude bull trout from streams. And so what we wanted to do is say, well, how do brook trout mediate the response that bull trout will have to climate change? How are they shaping the distributions right now, and how will they in the future? So for this work, we focused on 630 sampling locations in the Upper Clark Fork Basin in western Montana. So this is a zoomed-in view of that study area. Each of the individual points here are sampling locations. The white points are places where we did not detect bull trout DNA, and the black points are places where we did detect bull trout DNA. And then you spend 2 weeks in the lab, and you can generate information on the distribution of brook trout directly overlaying this. So here I'm using colors to indicate the concentration of brook trout DNA. So gray is no brook trout DNA, and then yellow to red are increasing eDNA concentrations. And eDNA concentration is relevant in this system because previously found the eDNA concentration in the environment here on the x-axis is positively correlated with eDNA concentration on the y-axis. And from hearing some of the talks before, I think that people are incredulous of these kinds of relationships, probably with good reason. So I'm going to just take 1 minute to talk about data quality. I'm not going to talk a whole lot about our specific methodologies, but I can provide more detail on that in conversation, or I can send you protocols. But we were able in this study to cross-reference with historical electrofishing sites about, I think it was 48 sites. And so we had paired within 1 kilometer electrofishing data and eDNA data specific to this basin. And we had greater than 90% concordance for both bull trout and brook trout. So we detected bull trout and brook trout at every single site where they had historically been detected, and we just picked them up at a couple more sites. And where that happened is where we picked them up with eDNA just a little bit upstream for brook trout. And so we have a 100% detection rate, and those places where we pick them up additionally with eDNA make sense biologically. And then this relationship, unfortunately I don't have a figure right now, but we were also able to correlate eDNA concentration with the number of brook trout that were captured with a single pass electrofishing reach at those sites, and there was still this strong linear correlation between the two. So based off of that, we feel that we're using eDNA concentration as a reasonable index of brook trout abundance in the system. Okay, and then because of the design of the bull trout inventory, the bull trout eDNA project, and now the design of the eDNA atlas, each of our sampling locations can be tied to the National Hydrography data set. So this gives us high resolution estimates of things like mean summer flow, mean August stream temperature, and stream gradient, which all have previously been found to be important in shaping the distribution of both bull trout and brook trout on the landscape. And then by working with colleagues in Boise with the U.S. Forest Service, we also have predictions of how these environmental conditions might change in the future due to climate change. So altogether, we have information on the distribution of bull trout, of their competitor brook trout at high resolution. We have information on the environmental context in which those species interactions are occurring, and estimates of how those are going to change in the future due to climate change, which sets us up really well to answer our questions that we have about bull trout and their future. So the purposes of this talk, I'm going to basically skip over all of the quantitative modeling that we did, and I want to just kind of show you the story that I think we discovered with images of the data. I'm happy to talk about the details of quantitative modeling, and then I also had the good news just yesterday that this paper will be out in Ecosphere probably in the next couple of weeks, and so you can go with more details there as well. So I'm going to use this data presentation a couple of times, so I'll take a minute to introduce it now. Here we're showing each of the sampling locations. These are the black points plotted in environmental space. So on the x-axis is mean summer stream temperature, on the y-axis is mean summer flow. And we're just using these colors to indicate the density of these points. So blue is a low density of sites, red is a high density. So now I'm going to use this same plotting to show you where we actually detected bull trout DNA. So again, each of these black points is a place where we detected just bull trout DNA. Now we're using the colors to show a relative density of points. So that is are the detections of bull trout overrepresented or underrepresented relative to the habitat that we sampled? So you can see based off the distribution of these colors that we have bull trout detections being overrepresented in slightly warmer and slightly larger flow habitats, and underrepresented in these slightly cooler and low flow habitats. So I'm putting a little ellipse around this region of overrepresented space and ask what happens when we add brook trout into the system. You can see there's this shift where now bull trout are associated with somewhat cooler and higher flow habitats than they were when brook trout had not invaded. And what this suggests is that potentially brook trout are competitively excluding bull trout from small stream habitats. And that's exactly what we find when we go to our quantitative modeling. It supports that conclusion, and it's consistent with our understanding of the biology of these species, where brook trout are a small stream specialist and bull trout are a large stream specialist. And previous studies have found that brook trout tend to competitively exclude bull trout from small stream habitats. So now the next piece to add on to this is climate change. So again, we're going to put a little ellipse around this region of overrepresented environmental space, which may represent habitat that is more resistant to brook trout invasion when we're talking about bull trout persistence. And we're going to keep this ellipse here and look at all of the sampling locations that we had. Again, this is under historical conditions. So this is about what these streams look like in terms of base flow and summer stream temperatures right now. But now let's look at what's predicted to happen in the future. This is 2040. You can see that there's this shift. Our sites are predicted to become warmer, and base flows are predicted to decline. And by 2080, we're expecting for most of these high flow sites to be completely thermally unsuitable for juvenile bull trout. And those few sites that, or excuse me, there's not that few, but for the sites that are cold enough for juvenile bull trout, they tend to be really low flow and potentially more susceptible to brook trout invasion impacts. So the immediate consequence of this is that large but cold streams that do remain on the landscape, although rare, may play a key role in the future distribution of bull trout. So I think the best way to illustrate this is by going to a map of the study area again. So here I've colored those habitats which are predicted to be thermally suitable for juvenile bull trout in blue and red. But red habitats are those that are not just cold, but also large. They're greater than 10 cubic feet per second base flow. So I shall go back and give you, for context, that dotted line there is 10 cubic feet. And let's focus here on a subbasin, the Little Blackfoot. So here we detected a low-density population of bull trout, and this was confirmed by repeated sampling efforts over several months and over space, despite the fact that we have extensive invasion of this system by brook trout. And this may at least in part be due to the fact that we have these large but cold habitats, and may be a kind of refuge from brook trout for bull trout. But if we look at what's predicted to happen in the future due to climate change, we're expecting for the loss of these habitats as base flows decline and temperatures increase. Whereas there are other parts of the basin which are predicted to stay large and cold longer into the future, and may represent a kind of refuge against both biotic invaders and climate change. And so I think when I've talked to managers in the National Forest, a lot of this actually is on National Forest lands, this has been really useful for them to understand how eDNA can be useful, because it gives them kind of a high resolution perspective of where they can make strategic management action. When you think about things like trying to stem invaders, or you're trying to do habitat restoration. But more broadly, I think that's kind of a neat example of how it's important that we incorporate biotic interactions into our predictions of how communities and species are going to respond to climate change. So basically, we've been predicting that bull trout are going to track their climatic niche upstream into cold, but that means small streams in the future due to climate change. But this study indicates that brook trout may competitively exclude bull trout from these habitats. And if we fail to include brook trout, we're missing a big picture of the future, a big part of the picture for bull trout looking into the future. All right, so the reason that I'm kind of illustrating this case study is that I think that it's an example of how new inferences can be pretty cheap and easy to produce when we're being intentional about how we archive our samples and our data. And so for the last part of the talk, I just want to walk through some ideas that I've had kind of specific to the National Genomics Center, but I hope are a little bit more broadly applicable about how we can do data and sample archiving effectively in the future. So there are a few simplifications with the current model for the eDNA Atlas project. One is that we're using the same standardized protocol for all of our sample collection. Another is that all of the analyses are actually be done at the National Genomics Center. There's one contributing eDNA lab. And then the last is that we're only using data from taxon-specific hydrolysis assays, qPCR assays, that have been extensively validated. We don't host things like metabarcoding data at this time. In the future, we want to build the capacity to incorporate data from multiple sampling designs. As Caren pointed out really well, there's not just one protocol that's going to be appropriate for all systems and all questions. We want to be able to incorporate data from multiple eDNA labs. So we don't want this to just be a National Genomics Center effort. We want this to be a larger community effort with multiple labs contributing their data. And we want to build a capacity to host more complex data types. So things like metabarcoding, where you have sequence generation, you have bioinformatic inference, you have comparison with a reference library to actually come up with your inference of species detection or non-detection. And so to get at these things, I think there's 4 things that we really need to focus on building into this kind of database and archiving structure of the future. So I'll walk through those bullet points, and again, I'll try and build up a kind of a cartoon of how this might be structured in parallel with that. So these are the first 2 points, and I've already talked about them a little bit already. That analysis results should be accessible, and that the physical storage is something that we need to think carefully about as we move forward. I would like to advance this idea that the null for eDNA analysis should be public release of data, because that benefits everyone. That when you make those data publicly available, then others can go in and not just use those data directly, but they can understand where they can complement existing sampling efforts. So that actually increases the quality and the value of the initial sampling effort. And it also provides opportunities for folks to revisit each other's samples and do new analyses at a fraction of the cost of doing a completely new sampling effort. And Caren Goldberg and others have brought this up really well, that there are some special cases where you don't want to provide specific geographic coordinates for a sensitive species that could end up having them targeted for pet collectors or poachers. And so we certainly want to protect the resources that we're surveying, but I think that in other cases we should be thinking about how can we deal with our eDNA processes in a transparent way. And I think one other point that I would argue on this is that I don't think that we're decreasing the quality of the data that we're providing to our collaborators have very specific questions by doing this. And if anything, we may be providing them with a product that has additional credibility tied to it, because we've committed to saying that we are going to fully describe how we collected that sample, how we did the analysis, and whatever those results are, we're going to make them available. And so when you launch an eDNA project and you're going to regulators, you're going to collaborators, there's this level of commitment to transparency that I think can be really valuable. And then the other one is physical storage. And I don't have an immediate solution for this because right now we have a basement full of freezers, and that's where our samples are kept. But ideally, this is an archive that we maintain long into the future. This DNA can stay around for a long time, and we need to be able to ensure the integrity of those samples, both protecting them from degradation and ensuring that they don't become contaminated in the future so that future researchers can use these because they're going to have new questions and they're going to have new technologies. And so I want to protect the interests of scientists that are smarter and better equipped than I am in the future. And this isn't impossible to do. Certainly we have biological archives of all different kinds which are well curated, but we need to get some critical mass behind this in order to, to build the infrastructure. So this is the backbone of kind of my idea for what this might look like. So I've already described this, and this is what we tried to put in with our first kind of shot at the eDNA Atlas, a public database, and then this physical or in vitro archive that we can revisit and populate the atlas with. Okay, and then the next piece is to incorporate complex data types. So what I mean is with metabarcoding, you have a more complex detection process. First, you're doing an enrichment step in the lab, then you're generating sequence data, and then that sequence data is interpreted through a bioinformatic pipeline in comparison with a reference library. And so we can be sure that bioinformatic pipelines are going to improve in the future, and certainly our reference libraries are going to improve in the future. And so I would argue that we need to be careful that we're archiving our raw sequence data and tying it to the original sample collection so that others are able to go back and access those raw sequence data, do a new analysis with their improved bioinformatic pipelines or with different bioinformatic thresholds, which may be more appropriate for certain questions, and comparing them with their reference libraries. Again, feeding into this eDNA Atlas. And I say the Atlas, but I just mean a public database. It doesn't have to be the National Genomics Center version of this. And then finally, the piece that holds all this together is comprehensive metadata. That is, we need for someone who accesses this public database to be able to understand how the sample is collected, how the laboratory analyses were conducted, and then how you came up with your inference from those laboratory analyses, perhaps through a bioinformatic pipeline. And this is particularly important if we're going to be drawing samples and data from across different eDNA labs and across different projects and over time. Because you might have a sample that was collected historically, you need to understand how that sample is collected so that you can interpret your laboratory analysis. Or if you're going to do a new bioinformatic working from the in silico archive, you need to understand how those sequence data were actually generated in the lab. And so this is where standardization comes in. In order to express the variance in ways in which you might collect, analyze, and interpret an eDNA sample and an analysis, we need to be able to agree as a community as to what the bounds of variance can be so that we can express that within a relational database. And if we can do that, then folks will be able to go into the database and filter based off of some criterion that they have about how the samples were collected or how the laboratory analyses were conducted. Or they have the ability to go in and use that metadata in their statistical framework formally and account for it that way. And so that brings us kind of, again, to the theme of this workshop, and part of the reason that I've been excited about it is that standardization really is what holds all this together, and it's what makes data and sample sharing across labs possible. And I know that I can be someone who's tempted to think that when I hear the word standardization, that it's going to be a pretty boring conversation about minutia, and that we're going to just be talking about which filter membrane you should be using. We're going to be talking about number of PCR replicates for hours and hours. But it's not just about minutia. I really think that it lays the groundwork for working as an eDNA community and accessing more of the biodiversity information from our samples. And if we can get that standardization kind of framework in place, then we can start moving from our kind of species by species, region by region eDNA survey efforts to piecing together more spatially comprehensive views of species distributions by combining different projects, by revisiting our samples and creating a greater taxonomic view of the biodiversity that's captured within our environmental samples, and by building up this database over time so that we have a temporal understanding of how these communities are changing. So that I really think that's, as an eDNA community, more than any one technological advancement, that's going to allow us to piece together a more complete image of biodiversity in the short term. And that's all I have. So I'll take any questions. Yeah, Bob.
Thank you for a great talk. I really liked the idea at the end about the metadata conversation. Have you guys started to actually think about metadata ontology, linking to Dublin Core, Darwin Core, the biological collections ontology, and some of these things that have been meant to help standardize our lexicon, both within our discipline but across biological science?
Yeah. So Bob's question was about if we, how we've, I'm not sure if I may be able to summarize it as well as you said, but if we've thought about how we're going to think about metadata, if there's other models that we can go to and tie to when we're doing this. And the short answer is that we're really early in this process. So starting to first, we just released the eDNA Atlas, I think about a month ago, and so starting to just kind of connect with other eDNA researchers and ask, what might this look like? So I really am interested in hearing more from you and more from others here about what that could look like. It does remind me of a point that I wanted to make here, and I think it relates to some of what other folks are talking about. Is that I think I would advocate for when I'm thinking about this database, the idea that standardization doesn't necessarily mean you must do it my way. It just means when you do it, you have to be able to fully and explicitly describe how you did it, so that anyone, even after you're dead, can come back and understand those data. And that's the structure around which we're trying to build the eDNA Atlas. Yeah, Caren first.
Really cool stuff. Just like one thing to consider, like in our deliberations and stuff like that, towards standardization, is the framework of like the minimum information required for an eDNA experiment kind of idea.
Yeah.
Just like we have those types of ideas for qPCR experiments or DNA array experiments.
Right.
I think that's kind of where we're going.
Yeah, yeah. And so to rephrase that that comment of it, is that it's important that we think about how we can set minimum information criterion for eDNA experiments broadly, not just for assays, but across all of the processes that occur there. And I think that that's a great point. I think that Caren kind of touched on this with the efforts that she's made about with trying to get people together and decide on solid criterion, right? There are some places where you're going to be able to solidify the variance down to like you have to do this, you have to use bleach, or you have to use UV, or you have to have separate spaces. And then there's other places where there's going to be it's going to be very legitimate to have high levels of variance. But we can't have so much variance that we can't compress this into an understandable database structure if we're going to share data across labs. So that'll be an interesting conversation to see what places we can squash into zero variance and what places we need to leave a lot of latitude by system and study. And I'm sorry, there was another question here, please.
I'm interested in the idea of physical archiving.
Yeah.
Because right now you're looking at bull trout and brook trout.
Yep.
But in fact, you could characterize human pathogens, you could characterize animal pathogens, plant pathogens.
That's true.
So when somebody gives you a sample, is there any thought or discussion of the ethical, legal, or political ramifications of what those samples can do?
Yeah, we're working through that right now. Yeah, I thought about that. The first point I brought up before the eDNA analysis, I was like, well, human DNA. There's human DNA. Are we going to identify individuals? Because I work with wildlife folks who do camera trapping. I'm sorry, I didn't rephrase the question. The question was, are there ethical, legal implications to the fact that you could reanalyze these samples for anything, not just bull trout and brook trout, potentially pathogens are the ones you brought up. So folks weren't too concerned about individual human ID. Just because that technology doesn't exist now doesn't mean it won't in the future. We're not, I can just say right now that we're not hoping to do that in the future. But yeah, pathogens are more complex. So right now we're talking with collaborators about saying, what we said to them is we want to make that data publicly available if it's appropriate to do so and have your permission. And I think the way that we're structured right now is that we will go to you for permission before someone re-accesses your sample and does a new analysis on it. And part of that is because our collaborators are thinking, well, maybe I want to do one more analysis on that sample. So they want an aliquot left. They're not thinking as big picture. But that gives us, that helps, I think, to some extent our collaborators not feel like they're giving this data to us and then we can do whatever we want. But generally, eDNA is a massively democratizing force in that sense, right? I mean, right now we're talking about standardization in part because anyone can go out there. Anyone who has a thermocycler can do eDNA, and no one's controlling who can take a water sample. So, yeah, if anything, I would say this is a safer place for us to have our eDNA samples because we're going to think about it before we just run some pathogen up in it. But anyone can mess with these things. Does that answer your question? Yeah, Bob.
Just a comment to that point. Museums, particularly like the American Museum of Natural History, where we set up a cryo collection for environmental samples, already have well thought out collection protocols, biomaterial transfer agreements. We don't have to start from ground zero on that. But usually institutions will not agree to archive samples if they don't have some latitude to use them in the future. Unless you're going to pay to store them like you would in the case of some of these patentable microorganisms that are part of an industrial process, that you pay to have them archived at the American Type Culture Collection and no one else can touch them because they're part of your patent application.
Yeah, and that's a great point. So Bob brought up the fact that we don't need to build this archiving structure from scratch by any means. And I think when we've talked about it, we say, well, our ideal situation would be that some archivists would come along, who have a facility, and want to take our samples. If the Smithsonian wanted to take this on, then that would be great because like I said, we have a basement full of freezers right now. We don't have a trained archivist. We're doing our best to be intentional about this, but yeah, we don't need to reinvent the wheel on this. And I also agree, Bob brought up this point about saying that, generally when you accept a sample into your archive, you are asking to have some freedom to do reanalyses on those samples. And I do want to push towards that as well because there's a lot of benefit to sending us your sample. You get your data put up on the eDNA atlas. It's tied now to the National Hydrography data set. You can integrate your samples with a whole bunch of other ones at the click of a mouse. You can call us up and say you want your aliquot from years and years ago. You're getting a lot of benefit out of doing that, and I think part of that should be contributing as part of the scientific community. You should be making your samples available for other legitimate studies as well. Great, well, oh yeah, Louis.
So we're playing with a very tiny amount of DNA, so I'm just thinking like, it's a great idea, the archiving, but it's not like, other types of things that you may archive, you may run out of material very quickly if you share samples. Is that, the long-term planning, is that a concern?
Yeah, so there's a few things. So one idea is that the in silico, kind of the sequence base, is we've played around this idea of like, well, what if you can create a digital representation of the biodiversity within that sample? So there's a whole bunch of limitations to that, right? But often, for example, our metabarcoding efforts are, and sometimes we feel constrained by reference libraries. But if we have this belief that our reference libraries are going to be more complete in the future, we can amplify and sequence and store those sequence data and expect that we're going to have more complete reference libraries in the future. And so that can be kind of an additional way in which we're archiving. You could be doing a specific metabarcoding study already, but you could also be doing metabarcoding regularly on your samples to capture some of that biodiversity information. But it is challenging. We've thought about encapsulation of DNA. So you don't need to use freezers. But I'm certainly open to ideas about how do you deal with these limited samples? Yeah, Bob.
On that end of, all samples, whether they're tissue samples in your freezer, they're all finite. This is not a new problem. Typically, just coming back from my museum background, you'd have a curatorial oversight committee that would review a proposal to use a specimen and have to weigh the impact of the study against its consumption to decide whether or not it was an appropriate use of that material. One other thing that I think is terra incognita, but we've played a little bit within the lab, this idea of biological reference standards, even for things like food authentication. You need some positive controls for commercial labs to run. That's part of where we run into trouble with FDA made its own tissue library for fish ID, but the commercial testing community can't get access to those because FDA says, look, we've only got one little slab of tissue and we have to run our own positive controls. We can't give it out to the community. In this case, we took tissue samples and extracted genomic DNA, exposed them to whole genome amplification, and then genetically analyzed them. And that WGA process had perfect fidelity. In the case of barcoding, we got good recovery of good sequences, so we could make a synthetic representation of a standard reference material that could be used as a positive control or in proficiency testing of commercial labs or other things. No one, to my knowledge, has yet even thought about, could we do some sort of a whole genome amplification of eDNA? But it may also be possible in cases of really rare and important samples that you might be able to actually synthetically replicate them.
Yeah, and Bob brought up a really good point about whole genome amplification as being a way of basically just increasing the amount of template that's there. And that's something we have been thinking about, which is, can you just archive that? And then once you have a lot of DNA to deal with, it lasts longer, and it's easier to do things like silicon encapsulation. That's what folks are doing right now, is taking synthetic DNA and encapsulating it. But you need more DNA than we have in our environmental samples. So that's a great idea. And if anyone here has lots of experience with whole genome amplification, particularly in complex samples, I'd be interested in talking about that, about whether and to what extent there's any bias that's introduced when you do that. Of course, that means more PCR products in the lab, which I dislike. But
A different lab.
A different lab, right? Yeah.
Terrific. Thank you. All right, for those of you with keen olfactory perception, you'll recognize that lunch is here. So I would invite you to form a line on either side of the buffet so that we can keep things moving relatively quickly. Or are you guys actually serving? Do you want one line or two? Just one. Okay, one line. Sorry, I take that back. But we have servers to help facilitate that, so let's have lunch.